Hello boys, welcome back to another session of online biology video tutorials. This session is meant for class 12th biology students and this is meant for 2nd July 2021. We have been discussing about chapter number 2, that is sexual reproduction in flowering plants. As you all may recall that we were talking about the reproductive part of the plant, that is flower. And why is it so? Why is it so called? Because of the presence of two reproductive structures, namely androecium and gynoecium. And we were also talking about the microsporogenesis as well as megasporogenesis. Now, once functional megaspore is formed, pollen grains are formed the other side. Now the question is how the pollen grains fuse with ovum. In particular, we can say nucleus of the pollen grain should be fusing or should be united with the nucleus of the ovum so as to form zygote. This process of formation of zygote is fertilization or syngamy. To happen this, there has to be pollination. That is, we said transfer of pollen grains from anther to stigma. If it is same flower, then we say self-pollination. On the other hand, pollen grains from the anther reach the stigma of another flower. That is, from flower 1, it's going to flower 2. Either say same plant or a different plant. Then we say cross-pollination. We also gave technical words for this like autogamy, xenogamy. Then we also talked about the various constraints for self-pollination, all that. So basically, our effort should be to avoid as far as possible self-pollination. The reason being, I said, there will be no varieties in the nature. More the cross-pollination, more the varieties, because genetic pool or genome of one plant will be mixed with genome of the another, leading to variable genomes. So presence of variable genomes will lead to variable morphological as well as anatomical features. So once a plant gets anatomical and morphological differences compared to its ancestor, then we say it is a variety. Hence, the aim of the cross-pollination is to make varieties. So what are the outbreeding devices that we can plan? 1. Hermaphrodite flowers can undergo self-pollination, that is autogamy. A flower having both anther as well as ovary, that is hermaphrodite flower. Continued self-pollination results in inbreeding depression, as I said. Continuously, generations after generation, say 5 to 6 generations, if there is a self-pollination, then the genome will lose its vigor. So the future generations will not be having so much of better qualities or high qualities compared to ancestors. This we call it as inbreeding depression. To avoid self-pollination and encourage cross-pollination, there are some devices in the plants. So plants themselves will try to make cross-pollination. So what is that? First one, avoiding synchronization. Yesterday we said pistil pollen synchronization. If we can avoid that, the plant has no choice but to go for cross-pollination. Here, the pollen is released before the stigma becomes receptive, or stigma becomes receptive before the release of the pollen, so that self-pollination can be avoided. Say anther, this is not matured, but already this is matured. So even if the matured pollen grains fall on the stigma, but the stigma is not ready, hence no self-pollination. Reverse. The ovum is ready, it is matured, but the pollen grains are not matured. This is not yet matured. So there is no question of self-pollination. B. Arrangement of anther and stigma at different positions. They will be arranged in such a way that the pollen grains may not be able to fall on the stigma in spite of having pollinating agents. So different positions. Then C. Self-incompatibility. We said compatibility of pistil pollen. Now incompatibility, opposite to that. It is a genetic mechanism to prevent self-pollen, that is, pollen from the same flower or other flowers of the same plant, from fertilization by inhibiting pollen germination or pollen tube growth in the pistil. Okay, pollen falls on the stigma, but once it reaches the stigma and falls, this has to germinate, and it has to germinate and form one pollen tube within the style. So there may not be enzymes which will make these pollen grains to germinate. So there is no question of pollen tube being formed. Hence, incompatibility. Next, outbreeding devices. Production of unisexual flowers. If male and female flowers are present on the same plant, that is monoecious, we say. Monoecious is a plant having both male as well as female flowers on the same plant. Example, castor and maize. Look at this, castor and maize. It prevents autogamy but not zethanogamy. So, since it has got both male and female flowers, the monoecious plant, it can prevent definitely autogamy. Look at that. This is in castor plant. This is how a female flower will be there. This is how a male flower will be there. Remember I told you, flower means don't think of feminism. There are male flowers. Okay. And similarly maize, you can see the male flower and the female flower. So such plants are called monoecious. And if male and female flowers are present on the same plant, that is monoecious, like castor and maize, it prevents autogamy but not zethanogamy. In dioecious plants, on the other hand, like papaya has got two types of plants. Male papaya, you must have seen in your own home, female papaya. The papaya plant, male and female flowers are present on different plants. So we say dioecious, male papaya plant, female plant. This prevents both autogamy as well as zethanogamy. So it has to have xenogamy only. The next, pollen-pistil interaction. What do you mean by this pollen-pistil interaction? The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil, which is mediated by chemical components of the pollen interacting with that of pistil. This we call it as pollen-pistil interaction. So basically it is a process in which pistil recognizes the compatibility or incompatibility of the pollen through chemical components produced by them, basically either enzymes or hormones. If the pollen is compatible, that is the right type, the pistil accepts it and promotes post-pollination events. Okay, and pollen grain germinates on stigma to produce a pollen tube. Look at this. This is the many pollens. One of the pollens started germinating because there is interaction, there is compatibility. Now this has got pollen tube getting germinated. So once the pollen tube is getting germinated means there is pollen pistil compatibility. And thanks to the chemicals that are present in the stigma as well as style region. And pollen grains germinate on stigma to produce pollen tube through one of the germ pores. Okay? The contents of the pollen grain move into pollen tube, basically two nuclei. So pollen tube grows through the tissues of the stigma and stale and reach the ovary. Look at this. It is reaching the ovary. And at that place, egg apparatus will be there. So it is ready. So A, pollen grains germinating on the stigma. Look at this here, the photo. And second, pollen tubes growing through the stale. See how many pollen tubes? Of course, you will be doing a practicals also here about the pollen germination. You all will see yourself with your own eyes, pollen tube germination. Now, continuing with this pollen pistil interaction. If the pollen is incompatible, see if it is compatible, there is pollen tube. The pistil rejects pollen by preventing pollen germination or pollen tube growth. Then we say incompatibility. In plants which shed pollen grains at two-celled condition, that is vegetative cell and generative cell. Remember, each pollen grain will have two cells, one vegetative big cell, one small generative cell. The generative cell divides into two male gametes during pollen tube growth. That's what I said. Male nucleus 1, male nucleus 2. Look at this. The germinating tube, now the pollen tube. Now inside the ovary you have got this antipodals, polar nuclei, egg cell and synergids. Now remember the structure, embryo sac we said 8 nucleate 7 celled. So remember that? Our 2 days back I explained to you. And basically here you can see the male gametes here 1 and 2. And of course there is a vegetative nucleus. And filiform apparatus is there which will facilitate the movement of the male nuclei. Synergids will be there, plasma membrane, of course egg nucleus and central cell. And what happens to this? Now the male gametes have already entered inside the ovarian apparatus, okay, through filiform. So male gametes are inside. And now the polar nuclei. Now what this male nucleus 1 will fuse with the egg. This is called fertilization. Now in plants which shed pollen in three-celled condition, pollen tubes carry two male gametes from the beginning. So if there is already three-celled one, then two male nucleus 1, male nucleus 2 will be entering inside. You can see here pollen grain on the stigma. Now germinating tube with two sperms. He says sperms, but I can call it as male nucleus 1 and male nucleus 2. Okay, and now there is a fusion. Here polar nuclei, egg apparatus. Now antipodals, polar nuclei. This is micropylar region. You have got two synergids and egg cell. Okay, and now here you can see the same one, the egg nucleus. This is important. This is the female part of it, and these are the male gametes of the male part of it. So there is a fusion. So pollen tube reaches the ovary, then enters the ovule through micropylar region. Look at this. This is what it is happening, micropylar region, and then enters one of the synergids through the filiform apparatus. Right. The filiform apparatus present at the micropylar part of the synergids guides the entry of pollen tube. So a plant breeder can manipulate pollen pistil interaction even in incompatible pollinations to get desired hybrids. This is what we do. That's why we say artificial. And then how do we do exactly this artificial hybridization? It is a crop improvement program in which desired pollen grains are used for pollination. This is achieved by following techniques. This also we will do in the practicals: emasculation and bagging. Okay, here you can see in the photos that the emasculation, removing of the male structure, masculine structure, and then bagging to maintain the genome so that there won't be any unnecessary outside interference with that. So in emasculation, what we do is removal of anthers from the bisexual flower bud of a female parent before the anther dehisces. You can see here it is being cut. The anthers are being cut. Emasculation is taking place. Now you can see here with the forceps. And on the other hand, bagging, here emasculated flowers are covered with a suitable bag made up of butter paper to prevent contamination. Look at this, contamination of its stigma with unwanted pollens. This is the purpose of bagging. When the stigma attains receptivity, matured pollen grains collected from anthers of the male parent are dusted on the stigma. Look at that here. Then the flowers are rebagged, as it is done here, and allowed to develop the fruits. So this is how we do artificial hybridization. For unisexual flowers, there is no need for emasculation. Female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower is rebagged. In unisexual flowers, we cover this, so there is no problem. So in our next session, we will discuss about a very important phenomena called double fertilization.